on? Oh, good. Hi. Hi, everyone. I'm Brenda Weigel. I'm the Division Director for Pediatric Hematology Oncology, and it is my pleasure to introduce one of my colleagues, Peter Gordon, who is an assistant professor in the Division of Pediatric Hematology Oncology, who came to us almost 2 years ago now from Harvard, where he had completed his fellowship work in pediatric hematology oncology and work in the lab of David Williams, and really focused on leukemia and cell signaling, and has been here continuing that work through NIH funding and through Leukemia Lymphoma Society funding, looking at new strategies for the treatment of leukemia. And so we're delighted today to have him share with us some of his very exciting work and a bit of an overview on things to come in the field of leukemia in childhood. So Peter, we look forward to your talk.
Can everyone hear me okay? Yeah. So thank you for the introduction and the invitation to speak today. I'm sort of divided my talk into, it's kind of loud, into two parts. For about the first third of the talk, I'm going to try to cover some leukemia basics. So I'm going to talk a little bit about diagnosis, epidemiology, risk stratification, and some very basic thoughts about therapy. And while doing so, what I really want to try to also convey is the critical role that I think that clinical trials and clinical research have played in improving the outcomes of pediatric leukemia patients. And then in the remainder of the talk, I'm going to switch gears a little bit, and I'm going to discuss what I think is an equally important role that basic research can play in improving leukemia outcomes. And to do that, I'm going to discuss two projects that my lab is currently working on. One that focuses on the leukemia microenvironment, and the second project that is trying to develop a new method for targeting both leukemia and other cancers using nanotechnology. So I thought I'd start with a patient, and this is a patient that many of you have probably seen in clinic, or at some point in your career you will see. This is a 5-year-old girl who is previously healthy. She comes in with a complaint of fever, fatigue, leg pain, and bruising that she says has been getting progressively worse over the last couple of weeks. On exam, she's moderately ill-appearing. She has scattered bruising and petechiae, and she appears quite pale. And furthermore, on exam, you can palpate a liver and a spleen. So a CBC is obtained, and it shows, a white count of 25,000 with 80% circulating blasts seen here in comparison to normal neutrophils, a hemoglobin of 6, and platelets of 19,000. So to sort of state the obvious, this picture based on the history, the exam, as well as the labs, is certainly highly suggestive for an acute leukemia. So I want to touch first on a little bit on leukemia epidemiology. It is the most common cancer in children. As you can see here, it narrowly beats out brain tumors. There are about 3,500 new diagnoses of leukemia each year in children and adolescents in the United States. And we can divide the leukemias into several subtypes, and this is shown here. As you can see, about 3/4 or 75% of pediatric leukemias are ALL, or acute lymphoblastic leukemia. 20% or so are AML, acute myeloid leukemia, and the remaining 5% is a combination of CML, so chronic myeloid leukemia, and other relatively rare myeloproliferative neoplasms. And this figure here just illustrates the rate of leukemia as a function of age, and there's one or two points I just want to make on this slide. First, for AML, which is shown in orange, you can see that there's a spike in the rate in infants less than 1 year, and then a relatively constant rate until older adulthood when it begins to rise again. And this is in contrast to what we see for ALL, shown in green, where you can see there's actually a quite large spike in the young children aged 2 to 6, and then a relatively slow decrease or modest decrease through adolescence until it sort of levels off. So just as the patient I presented illustrated, leukemia can present in many different ways. There can be symptoms related to marrow infiltration, so anemia, thrombocytopenia, and neutropenia. There can be symptoms related to leukocytosis, or in the presence of a high white count and a high blast count, the leukemia cells can begin to sludge in the vasculature, in the lungs, and in the CNS. There can be symptoms related to other organ infiltration, such as bone pain, which we saw in the patient I described, central nervous system symptoms, and hepatosplenomegaly. Finally, we can also see new children present with bleeding and coagulopathies, as well as tumor lysis syndrome. So to make the definitive diagnosis of leukemia requires a bone marrow biopsy and aspirate, since fundamentally leukemia is a disease of the bone marrow. And this just shows a bone marrow from a normal individual, and you can see that there's plenty of fat deposits, as well as that there's cells of varying sizes and shapes and stainings, which really correspond to the variety of hematopoietic progenitors that begin within the bone marrow. So white cell progenitors, red cell progenitors, platelet progenitors. And this is in contrast to the bone marrow that we see in a patient with newly diagnosed leukemia, where you can see that virtually the entire bone marrow is infiltrated by very monotonous appearing cells. And if we look at these cells at higher power, you can see that they have an appearance that is very characteristic of blasts. Now, by morphology alone, or by looking under the microscope alone, it can be very difficult, if not impossible, to distinguish between ALL and AML, and it's essentially impossible to distinguish between T-cell ALL versus B-cell ALL. So in order to make a more definitive diagnosis of leukemia, we use flow cytometry. And this allows us to look at the cell surface markers that are presented on the leukemia blasts, and by doing so and looking at the pattern of the leukemia cell markers, we can make a more definitive diagnosis of whether this is AML or ALL, or within ALL, whether it's a T lymphoid or B lymphoid derived leukemia. So having made the diagnosis of leukemia, there are a couple of therapeutic considerations that I just want to briefly touch upon. The first point I want to make is that therapy is unique to the leukemia type or even subtype, and the first big distinction is AML versus ALL. So AML therapy is generally of short duration on the order of a couple of months, but it's very high intensity. It's generally done entirely inpatient, and there's a fairly high rate of morbidity and mortality just related to the treatment itself. And this is in contrast to ALL, where the therapy is of longer duration, generally 2 to 3 years, depending on the protocol, and it's of less intensity. Some of it's done inpatient, but much of it can actually be done as an outpatient in the clinic. And for much of the therapy of ALL, children can go to school and sort of do the normal things in their lives. But even when you move beyond just the distinction between AML and AML, there are also going to be therapy differences. An example of this in ALL is we treat T-cell ALL slightly different than B-cell ALL. And historically, T-cell has been more difficult to treat, and they receive slightly more intensive therapy. The other important point that I want to make about therapy, and this is a general point, is the notion of risk stratification, or trying to balance efficacy and toxicity, or getting therapy just right. So providing enough therapy to cure the patient, but not providing excessive therapy to cause undue late effects. And so for risk stratification, we can think about a number of things that are both clinical and laboratory. We can look at patient characteristics, we can look at leukemia characteristics, and finally we can look at the response to initial therapy. And we can do this in the next couple of slides. I'm going to sort of review a couple of these basic ideas. And I'm going to do it in the context of ALL, but in fact, we use a very similar approach for AML. All right, so first, patient characteristics. And so these are things that we're going to know about the patient pretty much when they walk into the emergency department or shortly thereafter. And the first is age. 1 to 10 is considered to be most favorable. So we know that infants less than 1 have a very sort of unique form of ALL that can be very difficult to treat. White count less than 50,000 is considered favorable. Involvement of the central nervous system confers a more negative prognosis. Trisomy 21, I think, is sort of fascinating from the oncology perspective. So individuals with trisomy 21 have a significantly reduced risk of developing solid tumors, but a much higher risk of developing leukemia, both AML and ALL. And when they develop AML, their actually cure rates and survival is much better than the general population, whereas with ALL, they generally do slightly worse. And then finally is gender. And many protocols, boys are treated for 3 years, whereas girls are treated for 2 years. So in addition to patient characteristics, we can also look at characteristics of the actual leukemia itself. And the first distinction, and I've sort of already touched upon this, is just whether it's B-cell derived or T-cell derived. Again, historically, T-cell derived leukemias have been more difficult to treat and have required more intensive therapy. But we've become more sophisticated, and in addition to just distinguishing between whether it's B or T, we can also start to look at the genetics of the leukemia. So the first thing we can do is we can look at chromosome number. Is it hyperdiploid, so too many chromosomes, or too few chromosomes? Hyperdiploid usually confers a good prognosis, whereas hypodiploid is a more negative prognosis. Furthermore, there are a number of recurrent chromosomal translocations that we see in pediatric leukemias, and I've listed just 2 of them here. And again, they can confer a better or a worse prognosis. And one of the most common ones is the 1221 or TEL-AML1. But in addition to sort of recurrent translocations, there are other recurrent genomic alterations that we've identified that, again, carry prognostic value. Examples are amplifications of regions of chromosomes, deletions of individual genes or deletions of regions of chromosomes, and finally, point mutations that either activate or inactivate particular genes or proteins. And so this slide just illustrates the frequency which we can see some of these genomic alterations. And again, you can see that two of the most common is being hyperdiploid, so having too many chromosomes. About a quarter of children, and especially in the younger children, we see this, as well as the 1221 or tel-AML1 translocation. Again, occurs in about a quarter of kids, particularly in the younger kids. And again, these translocations or genetic alterations carry prognostic significance. You can see that patients with a 1221 or high hyperdiploidy do quite well, whereas patients who have too few chromosomes, so hypodiploid or have a 922 translocation or an amplification of a region of chromosome 21, do significantly worse. So now, in addition to the patient characteristics and the leukemia characteristics, we also look at response to initial therapy. And so for this, we really assess the response after the first couple of weeks of therapy. And for this, we use a test called minimal residual disease. And we can do this either by flow cytometry or by PCR. In either case, it's extremely sensitive. It can detect 1 in 1,000 to 1 in 10,000 leukemia cells within the bone marrow. And what we know is that the more leukemia that's still present after that first month of therapy, in general, the worst prognosis. And this is illustrated here. So in patients in this trial who were MRD negative, so who had less than 1 in 10,000 leukemia cells in their marrow at the end of that first month, had an excellent overall survival. But in patients who had even 1 in 10,000 to 1 in 1,000 leukemia cells in their marrow after that first month of therapy, you can see their prognosis is significantly worse. And as there's more disease after that first month, the prognosis continues to worsen. So really, let's come back to our patient now. So what we can do is we can combine these risk stratification factors to assign a final risk group. And this final risk group is then going to determine the intensity of therapy. And as I alluded to, ALL therapy generally lasts between 2 to 3 years. And so what I've listed here are sort of the generic components of ALL therapy, as well as the different drugs that we use during these different phases of chemotherapy. And again, the only point I want to really make with this is that the details of this are going to depend upon the final risk group, that some patients are going to receive more intensive therapy, depending on their risk stratification, whereas some patients are going to receive less intensive therapy. So to sort of come back to our patient, again, she's a girl. Her white count is favorable. She has pre-B cell leukemia. Her CNS was negative. She's of a favorable age. She has a favorable tel-AML1 translocation. She's MRD negative at the first month of therapy. So all of these are favorable characteristics and puts her into the low-risk category. And this really then allows us to answer the question that the parents really want to know, and that's what is her prognosis? And so I'm showing that here, and what you can see is that in the modern sort of era, a patient with low or average risk ALL actually has an excellent outcome, probably greater than 90%. But I think one of the interesting things that this slide illustrates is that in the not so distant past, her prognosis would have been significantly worse. And I think one of the interesting things this slide raises is sort of the question of, well, what's changed since the 60s and 70s in ALL therapy to get us from here all the way up to here? And I think one hypothesis, just looking at the data, you may say, well, maybe every 10 years or so, we develop some new great drug that makes a huge difference, and that's what these sort of jumps in outcomes are. However, actually, if you look at the leukemia drugs that I showed you on the previous slide, they're actually all really old, and they really haven't changed much. So I've listed here for ALL the drugs that are sort of the mainstays of therapy in the year when they were first identified. And then I did the same thing for AML here, which you can see most of these drugs have been around since the 50s or 60s. And so I don't think the answer to that question is that we have developed new exciting drugs that have changed sort of therapy. And I don't want to oversimplify things, but I think one of the major reasons that we've had such an improvement in outcomes in leukemia therapy, especially in ALL therapy, is because of successive clinical trials. And so these are large cooperative group trials with multi-institutions to get enough patients and often multinational. And it allows us to really compare treatments and make sort of iterative changes that's going to build on the success or occasionally the failure of the prior trial. And this really has allowed us to use chemotherapy more effectively, to combine the agents I showed you on the previous slide more effectively, and it has also allowed for the risk stratification that I just showed you. So it has allowed us to identify which patients need more therapy to be cured, and in which patients can we safely reduce therapy, still cure them, but reduce the long-term morbidity and mortality from treatment. And this here is a slide from the St. Jude Consortium that shows successive St. Jude trials in ALL therapy. And again, you can see this really impressive increase in outcomes from 10% to over 90% for a standard risk ALL patient. Okay, so I've already showed you this slide, which is just the survival curve for ALL. And I think it's interesting that it's remarkably similar to the curve for CML, in that this is now a highly curable, or at least treatable, I should say, disease, when in the not-so-distant past, it again had a pretty dismal prognosis. But unlike ALL, I think CML is really an example of an instance when basic research or lab research really transformed the therapy and outcomes of a disease. So the identification of the BCR-ABL protein shown here, followed 20 years later by the discovery of an inhibitor of the BCR-ABL protein, imatinib or Gleevec, again, has really changed CML from being a fatal disease to a treatable, if not curable, disease. And I think this really nicely highlights what I think is the important role that basic or lab research can also play in improving leukemia therapies. And so that sort of serves as my segue to the second part of the talk, where I'm going to focus more on basic research. But I think this slide, final slide of this section, also nicely illustrates that although we've made significant progress in the outcomes for AML and ALL, I think there's also significant work that remains to be done. And I think this is particularly true for AML, where even today in 2015, our cure rates are only probably about 55 to 60%. And that's despite maximally intensive cytotoxic chemotherapy and in many children, stem cell transplant. So I think we need new and novel approaches to treating leukemia. Certainly, the identification of many of the genetic mutations or alterations that drive leukemia development is raising new therapeutic possibilities, as many of these pathways or genes are either directly or indirectly druggable with new molecularly targeted therapies. An example of that is down here at the bottom, KIT, which is a receptor tyrosine kinase that plays an important role in hematopoiesis, but can also undergo activating mutations in a number of human cancers, including AML and GIstromal tumors. And there are a number of potent inhibitors of KIT that exist, and in fact, in the context of GIstromal tumors, they've proven to be remarkably effective. And so this is a patient before and after PET scan, and as you can see that following treatment with imatinib, there's almost complete resolution of the metabolic activity in this GIstromal tumor. And so really what I wanted to do was ask a very similar question for c-KIT mutant AML. Could we add a KIT inhibitor to the therapy of a patient with a KIT mutation and improve their outcomes? And again, we need to improve the outcomes in AML significantly. So to begin to ask this question, we did a relatively simple experiment. We took leukemia cells from a patient with a KIT mutation and put them into tissue culture in the presence or absence of a KIT targeting drug. So both imatinib and nilotinib effectively target KIT. And then after 48 or 72 hours, we measured viability. And what you can see is that both imatinib and nilotinib significantly decrease both the viability and proliferation of a c-KIT mutant AML cell line. So from a therapeutic perspective, this is an encouraging result. However, I think a very important caveat to this result is that this experiment was done in a plastic dish in a tissue culture incubator. And so this completely overlooks the complexity of the bone marrow microenvironment, which, as we talked about, is where leukemia develops, arises, and is maintained. And I think this slide is complicated, but I think it sort of nicely illustrates the complexity of the bone marrow microenvironment. So as you can see, there are many different cell types in the bone marrow. There are osteoblasts, osteoclasts, fat cells, fibroblasts, stromal cells, hematopoietic cells, the long list. All of these different cell types can release a variety of soluble factors, including cytokines, chemokines, and hormones. And in addition to these factors, there's also a complex extracellular matrix. And what has been shown is that all of these factors can interact with leukemia cells. And these interactions between these factors and the leukemia cells have been shown by many groups to often be to the advantage of the leukemia. In other words, these interactions can promote leukemia development, promote leukemia proliferation, and make these leukemia cells more resistant to therapy. So in order to more accurately try to mimic this in vivo environment in vitro, we again wanted to first look at soluble factors that are secreted by cells within the bone marrow microenvironment. And so in order to do this, we took bone marrow stromal cells, put them into tissue culture, and after 48 or 72 hours, we collected the media, which again is going to contain any of the soluble factors released by these stromal cells, and And then we resuspended our leukemia cells in this media in the absence or presence of drug. And again, what you can see is very similar to what we saw before. With regular media, these leukemia cells are quite sensitive to imatinib and nilotinib. In other words, they are basically dying in the presence of these drugs. However, in the presence of bone marrow conditioned media, so media that had been grown on top of bone marrow stromal cells, these leukemia cells essentially become resistant to therapy, meaning they're growing and proliferating quite happily despite the presence of these KIT-targeting drugs. So we went on to show then that these bone marrow stromal conditioned media is actually protecting the leukemia cells from apoptosis. And so in this experiment, we had leukemia cells in regular media shown here, and in the presence of nilotinib, you can see a significant increase in apoptosis shown by a Nexon staining. However, this effect is almost completely blocked in the presence of conditioned media. And we went on to show similar results using a caspase activity and by morphology. So this really sort of raises the question then, how are bone marrow stromal cells protecting leukemia cells? And so the first step was to ask, well, what proteins, so cytokines, chemokines, hormones, are secreted by these bone marrow cells? And then we wanted to ask, what are the ability of these individual factors to actually protect the leukemia cells? And so here's a list of initial candidate factors, and here's an example of some of the factors that we've tested. And what you can see is that GCSF as a single agent can almost completely recapitulate what we saw with bone marrow stromal conditioned media. In other words, in the presence of regular media plus GCSF, these KIT mutant leukemia cells become resistant to imatinib and nilotinib. So what is GCSF? It's a cytokine secreted by bone marrow stromal cells and other cells that can cause white blood cells to grow and proliferate. Many of you may come across it on the oncology rotation because it's something we use clinically to try to raise somebody's white count after giving chemotherapy. But in this case, what we went on to show is that it also turns on survival signals in the leukemia cells, making them resistant to drugs. And what the data here is simply showing is that this protection occurs at levels of GCSF that can occur actually in patients, suggesting this may actually be a relevant mechanism in the patient. So this also sort of raises a therapeutic opportunity, since we hypothesized that if we could block the GCSF receptor, we may be able to restore sensitivity to KIT inhibitors. And this is sort of what the schematic here shows. And actually the data really bore this out. So again, you can see that in regular media, the KIT mutant leukemia cells are again quite sensitive to imatinib. In bone marrow conditioned media, again, they become largely resistant to the effects of imatinib. However, if we add an antibody that blocks the GCSF receptor and prevents the binding of GCSF to the leukemia cells, we can restore sensitivity to imatinib. So a couple conclusions from what I've showed you so far. We showed that cytokines secreted by bone marrow cells can protect mutant AML from KIT inhibitors. One biological implication from this data is that mutant KIT signaling may not be required for leukemia maintenance within the appropriate microenvironment, such as the bone marrow, despite an abundance of data showing that KIT mutations are in fact required for leukemia development. But this also has clinical implications, as there was a recent clinical trial that added a KIT inhibitor, dasatinib, to standard chemotherapy in adult patients who were likely to harbor an activating KIT mutation. I don't know what the results of this study have shown yet, but certainly some of the data I just showed you certainly raises some concerns about how much efficacy is going to be added by the addition of a KIT inhibitor. So this is just one example of many, many, many published examples of the importance of the bone marrow microenvironment in leukemia development, maintenance, and response to therapy. But as I showed at the beginning of the talk, leukemia is a systemic disease, and it actually occupies many different niches or environments within the body. And I've listed here just a couple of the different tissues or organs where we can detect leukemia cells in a new patient. And as I showed you, every new patient receives a bone marrow biopsy, but every new patient also receives a lumbar puncture in order to assess for the presence of leukemia cells within the central nervous system, since we know that leukemia has a propensity to involve the central nervous system. So for the next couple of minutes, I want to sort of switch topics from talking about the bone marrow microenvironment in leukemia and actually talk about the central nervous system microenvironment in leukemia. So as shown here, it can be a major obstacle to cure. About half of patients will develop CNS leukemia without adequate therapy, and I'll talk in a minute about what adequate therapy is. Furthermore, despite what we consider adequate therapy, isolated CNS relapses can occur in 3 to 8% of patients. And so what this means is that the patient has completed all therapy, is deemed cured, but then has their leukemia come back in their central nervous system. But when we do a bone marrow, their bone marrow is negative. Furthermore, in some clinical trials, up to a third of all relapses can be isolated to the central nervous system. So in order to both prevent CNS development in patients who are negative initially, or to treat patients who do have CNS involvement, we can use a variety of things. We can use intrathecal chemo. So in this case, we do a lumbar puncture, we take out some CSF, and at the same time, we push in chemotherapy. We can give high-dose systemic chemotherapy that crosses the blood-brain barrier. And finally, in some children, we use cranial radiation. And while these things can be very effective in treating central nervous system leukemia, they can also cause many acute and late complications in children. And I've listed just a couple of them here: neurocognitive deficits, endocrine disorders, growth impairments, secondary cancers. And I think this picture illustrates this nicely. So these are actually identical twins at the age of 26. The twin here was diagnosed with pre-B-ALL at the age of 4. And as part of her therapy, she received intrathecal chemo, high-dose systemic chemo, and cranial radiation. And clearly, at age 26, she's a long-term survivor, but I think this picture also illustrates some of the late effects of therapy. So while I think we sort of all recognize the clinical importance of CNS leukemia, I would argue that the role of the CNS microenvironment or niche in leukemia is very understudied. And this is illustrated here. So I did a PubMed search for bone marrow microenvironment and leukemia, and it returned about 800 citations. I changed bone marrow microenvironment to central nervous system microenvironment, got back 7. And in fact, most of these are not that relevant when you actually look more closely. So some of the things that we're interested in my lab are: how do leukemia cells traffic to the central nervous system? How do they cross the blood-brain barrier? How do they cross the blood-CSF barrier? And what is the role of soluble factors as well as direct CNS cell and leukemia cell interactions in leukemia cell survival and proliferation and response or resistance to therapy? And ultimately, can we leverage a better understanding of CNS leukemia into therapy that's going to be both more efficacious as well as less toxic for our patients? And so in order to do this, we've taken a combined in vitro, so cell culture approach, as well as in vivo approach, and I'll show you data from both of those. For in vitro, we can do what we call co-culture. So we can grow CNS cells, so choroid plexus cells, meningeal cells, and astrocytes in tissue culture plates. We can add leukemia cells to those cells. And what we find is that the leukemia cells actually adhere quite well to these different CNS cell types. We can then co-culture the leukemia cells that have adhered to these CNS cells and start to ask questions about chemosensitivity, proliferation and quiescence. We can do gene expression profiling as well as other experiments. And an example of one of these experiments is shown here. So we can take leukemia cells growing in suspension and then versus CNS or leukemia cells that are grown adherent to the CNS cells, so in this case meningeal cells and astrocytes. Then we can add the drug cytarabine, which is one of the mainstay drugs in the treatment of CNS leukemia, and then we can assess leukemia viability. And what you can see is that when grown in suspension, leukemia cells are quite sensitive to Ara-C, as shown by the increase in percent of apoptosis. However, when these exact same leukemia cells are grown adherent to meningeal cells, they become largely resistant to the effects of Ara-C. And so one of the interests of the lab is better understanding the molecular and biological basis for this resistance to therapy. And that's another example of sort of what we can do in the tissue culture hood, is trying to better understand how do leukemia cells make it into the central nervous system to begin with. So in order to cross over the blood-CSF barriers, leukemia cells have to go through a fenestrated endothelium, but then they need to cross over choroid plexus cells that have tight junctions. In order to mimic this in vitro, we've developed a system where we can grow choroid plexus cells on a transwell membrane, add leukemia cells to one well of the plate. 48 to 72 hours later, we collect the cells on the other side of the well, and what we can find is that we can detect leukemia cells that in fact have crossed over these choroid plexus cells. And we're still working on some of the technical details of this assay, but we think this could be a very useful approach in vitro to begin to ask what are the genes and what are the pathways that are important for leukemia cells to migrate to the central nervous system. And just as we're doing these experiments in vitro, we can ask many of these same questions in vivo in the mouse. And so for these experiments, we can take these same human leukemia cells, inject them into the tail vein of an immunocompromised mouse, allow leukemia to develop, which usually in these mouse models takes anywhere from 4 to 8 weeks. We can then euthanize the mouse when they've developed systemic leukemia and isolate their femur or their brain and central nervous system, and from these different microenvironments, isolate the leukemia cells. So same leukemia cells are injected, but just from different microenvironments. And then we can start to ask questions about similarities and differences by using pathology, gene expression profiling, flow cytometry, and other experimental approaches. And an example of this is shown here. So this is just basic immunohistochemistry on a mouse 4 weeks after being tail vein injected with human leukemia cells. In this experiment, the leukemia cells are stained brown with a human CD19 antibody. And what you can see is that about a month after injection, the mouse's bone marrow is almost entirely effaced by these leukemia cells. But in addition to detecting the leukemia cells in the bone marrow, we can detect the leukemia cells adhered to the meninges of the mouse, as well as within the cerebral aqueduct. So again, in the mouse, we can recapitulate what we see in patients, in that not only do they have bone marrow involvement, but they also have central nervous system involvement. So in one experiment, what we've gone on to do is we've, again, isolated in a mouse that has developed leukemia, the brain and the femur, and isolated the leukemia cells from these different microenvironments. And then we did gene expression profiling to begin to ask what genes are differentially regulated between these different environments, and in hopes that it'll give us more understanding about CNS leukemia as well as leukemia within the bone marrow. And so I've shown just an example of some of the data here. So this is a heat map. So things that are red are upregulated, things that are green are downregulated. And what this is comparing is gene expression in the same leukemia cells, but that were grown in suspension, that were grown non-specifically adhered to a plastic plate on retronectin, or that were isolated from the CNS versus bone marrow of the same mouse. And what you can see is that there's clear gene expression differences within these leukemia cells, depending on whether they're residing within the central nervous system or the bone marrow of the mouse. And this is just, again, a representative list of some of the genes that we identified, but many of them are very biologically interesting, and we think that they may provide us new insights into CNS leukemia, but we may also be able to leverage these results into better therapies for CNS leukemia. And I'm going to give you one example of a gene here that we identified as being upregulated in the central nervous system that we think has the potential to be very interesting. This gene is called PBX1. And one of the reasons we're interested in it is it has a long history of known to be involved in blood development as well as leukemia. It's been shown to be involved in the self-renewal of blood stem cells by maintaining quiescence as well as by preserving their lymphoid potential. But interestingly, it's also involved in a translocation with TCF3 that we actually frequently see in pediatric ALL. And I think perhaps the most interesting thing about this translocation is that it's actually associated with increased CNS disease and CNS relapse in human patients. And so we've gone on to confirm the gene expression profiling results using qPCR, and what you can again see here is that in leukemia cells isolated from the CNS of a mouse, there's a significant upregulation in PBX1 expression relative to the exact same leukemia cells isolated from the bone marrow of the same mouse. And furthermore, we can detect an increased expression of PBX1 in leukemia cells that were grown adherent to choroid plexus cells. And so we currently have work in the lab underway to better understand what are the functional consequences of PBX1 upregulation, as well as to try to look at many of these other genes that we've identified. Again, with the hopes that we can ultimately leverage this into a better understanding of leukemia and better therapies. So for the last sort of section of the talk, I'm going to switch gears one more time. I'm going to talk about a sort of a nanotechnology project. And really what this sort of stemmed from was just a very basic observation that we need therapies that are both efficacious and less toxic. And I've listed here sort of a number of sort of novel therapies that have been developed in the last 10 or 15 years that I think sort of meet these criteria. And this includes molecularly targeted therapy. An example of this would be the imatinib that I discussed for CML. Antibody drug conjugates, where an antibody delivers a drug to a cancer cell, or sort of in a conceptually similar fashion, prodrug activation in cancer cells. The sort of notion of targeting multiple cancer pathways rather than a single cancer pathway. And finally, immune and cellular therapies. And one of the reasons we're sort of excited about this approach that I'm going to describe is that I think it really combines aspects of these first three therapies. And so nanotechnology, well, what is it? So let's start with that. So really it refers to things on the scale of 1 to about 100 nanometers. And so this is about the size of a protein to a virus, or you can think of it as being about 1,000,000 to, I guess, 100 million fold smaller than a tennis ball. So relatively small. And in fact, sort of in detail, what we are proposing to use is gold that has been conjugated or functionalized with oligonucleotides. And this is actually technology that was initially developed by Chad Merkin's lab, an inorganic chemist at Northwestern University. And he and others have shown a number of favorable properties for these oligonucleotide conjugated gold nanoparticles. And that includes high uptake into many different cell types, stability in biological environments, including when injected into a mouse, minimal toxicity, and very low immunogenicity. So one of the reasons we're sort of excited about this approach that I'm going to describe is that we think it's a very customizable approach to activate a molecularly targeted drug in a cancer cell. And furthermore, we think it's potentially applicable to any cancer for which a drug and a unique target can be identified. So I'm going to show you some data in the context of leukemia, but I think it could extend beyond leukemia. And so this schematic here shows a little bit in closer detail of exactly what we are making. So we begin with a molecule of gold, and to it we can attach an oligonucleotide via a thiol bond. So we can order a synthetic oligonucleotide that contains a sulfur, and it attaches with very easy chemistry to gold. And because we chemically synthesize this oligonucleotide, we can make it in any sequence that we choose. And so we can choose this sequence to be complementary to an mRNA that is specific to a cancer cell. So then in the second step, we anneal onto this first oligonucleotide a second shorter oligonucleotide that is conjugated at its end to a molecularly targeted drug. So now the important thing to note is that the only thing that's holding this drug conjugated oligonucleotide to the gold nanoparticle is Watson-Crick base pairing. And furthermore, we can put about 200 of these duplexes on each gold nanoparticle. All right, so how does this actually work? So as I alluded to earlier, these drug-conjugated gold nanoparticles are taken up very well by many cell types. However, in a cancer cell, when these gold nanoparticles encounter the cancer cell-specific mRNA, this mRNA can bind to its complementary sequence that's on the gold nanoparticle. And in doing so, it's going to displace the oligonucleotide, the drug-conjugated oligonucleotide from the gold nanoparticle, which will remove the drug from sequestration and allow it to bind to and inhibit its target enzyme. And the important thing is that this is a binary reaction. So the more cancer cell-specific mRNA present in the cell, the more drug-conjugated oligonucleotide that's going to be released. And so we think this is an approach to increase efficacy and minimize toxicity, as in normal cells that lack this cancer cell-specific mRNA, the drug-conjugated oligonucleotide should remain sequestered to the gold nanoparticle. And furthermore, in sort of a second step of this reaction, cellular nucleases can actually degrade the cancer cell-specific mRNA sort of basically by antisense technologies, and again, thus further deplete the cancer cell of an mRNA upon which its growth and proliferation is dependent upon. To sort of summarize, again, sort of why we think this could be both more effective and less toxic. Again, we know that and I'll show you some data for this, that there seems to be preferential uptake of leukemia cells, or of nanoparticles into leukemia cells relative to normal bone marrow cells. And in fact, it's been shown that in a cell that takes these particles up well, there can be over 500,000 to 1 million particles per cell. And furthermore, as I pointed out, each of these particles can carry 1 to 200 drugs on it. And furthermore, in the presence of the cancer cell-specific mRNA, these drugs should be released in the cancer cell. In contrast, in a normal blood cell, for example, shown here, A, there should be less particles taken up, and B, even if they're taken up, in the absence of the cancer cell-specific mRNA, there should be very little release of the drug from the nanoparticle. So again, we think this could be applicable for any cancer for which a drug and an mRNA target could be identified. So a couple of sort of general considerations for selecting a drug. It should be clinically relevant. It needs to be chemically amenable to conjugation to DNA. And furthermore, the conjugation of the drug to the DNA should hopefully not perturb the drug binding and efficacy. And then as a second point, we need to think about what mRNA target to select. So there are many mRNAs that are going to be unique to a cancer cell. You could begin, for example, with an overexpressed mRNA, and I'm going to show you data for the gene survivin, which is an mRNA that's overexpressed in many cancer types relative to normal differentiated cells. You could think about an alternatively cancer cell-specific spliced mRNA, or finally, as I've showed you earlier, the breakpoint sequence of a translocated gene is going to be truly a unique sequence to a cancer cell, and thus may represent a very attractable target for this technology. And I'm going to show you some data for the AML1-ETO translocation, which occurs with relatively high frequency in pediatric AML. So here's some data. So in this is just illustrating the selective uptake of the nanoparticles. So in this experiment, we mix the leukemia cells with normal bone marrow cells. We add nanoparticles, and the next day we can assess what percentage of the leukemia cells have taken up nanoparticles. And what you can see is that even with 100-fold, and we have data even going out to 1,000-fold excess of normal bone marrow cells, we see almost complete uptake within the leukemia cells. And then the data on this side of the slide is showing the selective release of the drug or oligonucleotide. And so for this experiment, we engineered normal cells to express the AMO1-ETO transgene under the expression of doxycycline. Furthermore, we made gold nanoparticles that rather than having drug-conjugated oligonucleotide, have a fluorophore-conjugated nucleotide. Now, when this fluorophore-conjugated oligonucleotide is sequestered to the gold particle, the gold quenches the fluorescence, and so you don't see any fluorescence. However, if this fluorophore-conjugated oligonucleotide is displaced from the gold nanoparticle, it can fluoresce. And what you can see is that in the presence of the AMO1-ETO transcript, we see massive amounts of fluorescence corresponding to release of the oligonucleotide from the gold, whereas in the absence of the AMO1-ETO transcript, we see very little background release of the fluorophore-conjugated oligonucleotide. Again, suggesting that there's going to be selective release of the drug-conjugated oligonucleotide. So as sort of initial proof of principle, we started with the drug dasatinib. And we chose this because it's a very potent tyrosine kinase inhibitor. It inhibits SERC and KIT and BCR-ABL. It's being used and tested in both hematologic and solid cancers. And the other thing that was very attractive was that it contains this hydroxyl group here, which actually makes it quite amenable to conjugation. And this is shown here, and we did this in collaboration with Joe Piccirilli's lab at the University of Chicago. What we did is we converted the hydroxyl group to an azide. This azide can then very easily be reacted with any oligonucleotide that contains a terminal alkene group using a click reaction, and this is a commercially available oligonucleotide. So we can order an oligonucleotide of any sequence we want with a terminal alkyne and react it with our azide-modified dasatinib, and we get good yields and good efficiency with this reaction, and thus we're able to make dasatinib-conjugated oligonucleotides with relative ease. And the first thing we wanted to do was, in fact, confirm that we weren't perturbing the efficacy of dasatinib. And so to do this, we used a KIT and SERC in vitro kinase assay. And we tested both dasatinib as well as dasatinib conjugated to DNA oligo. And what you can see here is a comparison of the IC50s. And what this shows is that dasatinib conjugated to DNA is still very able to inhibit both SERC and KIT. So again, an encouraging result. We next tested it in vitro. So again, we used the SERC in vitro kinase assay. And in this experiment, we added the actual dasatinib survivin gold nanoparticles. And then we added an oligonucleotide or a control that mimicked the survivin mRNA sequence. And so if you start by just looking at this fourth column, so these reactions contain the enzyme SERC, and they contain the dasatinib survivin gold nanoparticles. And what you can see is that the dasatinib conjugated oligonucleotide remains sequestered to the gold nanoparticle, and SERC retains full activity, or essentially full activity. Now, if we add a scrambled DNA target, which is shown in the next lane, we essentially see the same thing. In other words, a scrambled DNA is unable to displace the dasatinib conjugated oligonucleotide from the gold particle, and again, SERC retains full activity. Now, in contrast, in this last column, if we add an oligonucleotide that mimics the sequence of the survivin mRNA, it can very effectively bind to the survivin gold nanoparticle, and in doing so, displace the dasatinib conjugated oligonucleotide, which then you can see here very effectively inhibits SERC activity. So another sort of encouraging result. So we've also begun to test this in cells. So in this experiment, we added our dasatinib survivin gold nanoparticles to leukemia cells. We allowed 24 hours to pass, and then we fixed the cells and stained them for phospho-SERC. And again, what you can see here in orange is that we can detect a very strong signal for phospho-SERC in the absence of any drug. However, if we add either dasatinib by itself, shown in blue, or our dasatinib survivin gold nanoparticles, we see very efficient inhibition of phospho-SERC activity. Again, suggesting that in vivo in the cells, these particles are behaving as we would expect. And we've also shown this for a number of other kinases that we would expect dasatinib to be targeting. So we've also begun to ask, do these efficiently target leukemia cells? And so to begin to ask this question, we started with 3 leukemia cell lines that we expected to be dasatinib sensitive. So Kasumi and SK101 both contain activating c-KIT mutations, and K562 contains a BCR-ABL translocation. And 32G cells were a control. It's a murine hematopoietic cell line. What you can see is that if we add our dasatinib gold nanoparticles to any of these three cell lines, we see very significant inhibition of cell viability and proliferation, whereas we see very little effect on our control cell line. And furthermore, this is just showing on the other side that this is a dose-dependent effect and that we see these effects at sub-nanomolar efficiencies, or concentrations, I should say. And finally, this is just illustrating that these effects on proliferation and viability are actually through an increase in apoptosis, as shown by an increase in annexin-positive staining. And again, we can see this effect at less than 1 nanomolar concentrations of drug. So in addition to being more efficient, I've hypothesized that this approach should be less toxic. And so to begin to sort of ask this question, we decided to select two systems where we know that dasatinib can inhibit the function of normal cells. And for this, we chose cell signaling in normal hematopoietic human CD34 cells, as well as T-cell activation, as there's been data showing dasatinib can impact both of these. And what you can see here is that for dasatinib by itself, it in fact does inhibit phospho-Circ signaling in the hematopoietic stem cells. But if we add an equal amount of our dasatinib DNA gold nanoparticles, we see essentially no effect on phospho-Circ signaling. And similarly, shown over here, you can see that dasatinib inhibits the activation of T cells when we add CD3 and CD28 beads. However, again, when we add our dasatinib conjugated gold nanoparticles, we see only a very small effect on T cell activations. In other words, the T cells can still activate almost like normal. So again, together, both of these suggest that, as we'd expect, we're seeing less toxicity. And I think this is a combination of two effects. First, these non-malignant cells, as I've shown, seem to take up these gold nanoparticles less well. And second of all, even when they take these nanoparticles up, the dasatinib conjugated oligonucleotides remain sequestered to the gold nanoparticles in these cells and thus can't have off-target effects. So just to wrap up, I've hopefully touched on a little bit of the clinical and basic research aspects of leukemia, and I think both have the potential to improve leukemia outcomes. I think understanding how leukemia cells interact with the many niches it's involved in, bone marrow, CNS, et cetera, may lead to new and less toxic therapies. And finally, I've shown some very preliminary data, but we're excited that we think these drug-conjugated gold nanoparticles may be a customizable approach to activate a drug in a cancer cell and thereby maximize efficacy and minimize toxicity. So I just want to conclude by thanking the people who've done this work. So Nathan, Jordan, and Ed have really done the majority of this work. The Piccirilli lab at the University of Chicago helped us with the conjugation of dasatinib to the gold nanoparticles. The pathology core has been very helpful with some of our immunohistochemistry. And finally, I'd like to thank all of the funding, and I'm happy to answer questions if anybody has any. Yeah.
Great review. Thanks, Peter. Is there a specific way to, I'm thinking about the complementation, how to get the nanoparticle to release the drug and bind tighter to your target as opposed to hanging on to the drug, or are you just relying on?
So we're relying on the presence of that cancer cell specific mRNA. And the more cancer cell specific mRNA, the more release of drug there's going to be. Does that make sense? Yeah.
Say that into the mic.
Just to follow up that question, you're saying this is a customizable approach. How do you go about identifying the mRNA that you're going to target for a particular cancer?
So that's a great question. Part of it is you can just start with gene expression profiling. For example, you can look at what genes are being overexpressed in your cancer of interest. So that's one way to go after it. Again, the breakpoint sequence of any translocation is going to be unique to a cancer cell. We know that there are many translocations that occur in cancer cells, and often these are drivers of cancer development. So they're ideal targets because they're important to the cancer cell and they're highly expressed in the cancer cell. Alternatively, we know that for a number of genes they're alternatively spliced in cancer cells, and so that splice junction again is going to be unique to those cancer cells. So really, you sort of just need to start with the cancer you're interested in and ask, do any of these categories apply? And I think for pretty much any cancer you can think of, you could come up with an mRNA that is going to be at least overexpressed relative to non-malignant cells. Mm-hmm.
There you go.
So, Peter, once you have the mRNA, you can target it there. But I don't think you also said that the leukemia cells selectively take up nanoparticles more so than normal cells. Why is that, and is that unique to leukemia, or do other cancer cells do that?
No, that's a great question. I can't give you the complete answer because it's something we're sort of actively looking at. But at least in our hands, it seems that most leukemia cells and most other sort of transformed cells that we've looked at seem to take these up very well. So we're starting to look at the mechanism by which they're taken up. And certainly we do see uptake into benign cells. And so I do think sort of the notion of only releasing the drug in the presence of the cancer cell-specific mRNA is important because clearly these things are going to be taken up by benign cells. The other things we've thought about and that could be done is you could actually further modify these gold nanoparticles with antibodies or other targeting agents to more selectively target cancer cells as well, to try to further increase the selective uptake within the cancer cells.
Me again, sorry. What sort of toxicities have you anticipated, and how will these be cleared and excreted? Is it going to rely on apoptosis of that cell that it gets taken up in?
So our data so far suggests that, yes, the cells where that are dasatinib sensitive and then dasatinib is released are undergoing apoptosis. In terms of toxicity, there are some previous papers that have looked at toxicity of oligonucleotide modified nanoparticles in mice and shown that they're actually quite non-toxic. So I think in the absence of drug, i.e. the dasatinib, these particles, at least in mice, are quite well tolerated, and certainly in other areas of medicine, there's examples of gold nanoparticles being used in patients. There was a clinical trial using gold nanoparticles to deliver TNF, for example, to tumors, and again, in the absence of the TNF, they're quite well tolerated. So I think the gold conjugated oligonucleotides by themselves actually exhibit very low toxicity, which is, again, one of the advantages of the approach. Cool. Thank you.